This video explains the cycles of glycolysis, cellular respiration, and also fermentation. Harvesting of energy from glucose has three stages: glycolysis, the citric acid cycle, or you can say Krebs cycle, and finally oxidative phosphorylation. This diagram summarizes the entire process of with all the cycles inside, beginning with glucose and ending with ATP. Take a look at the light blue box there. This is showing glycolysis in one single step, going from glucose to pyruvate. But actually, it could be broken down into several more steps, which I will want you to know. Once pyruvate has been formed, pyruvate can enter the mitochondria and enter the pyruvate oxidation steps, and then the products of pyruvate oxidation can enter the citric acid cycle, and finally the products of the citric acid cycle can enter oxidative phosphorylation, which consists of the electron transport chain and chemiosmosis. While we go through all these steps, it's important to keep track of how many of each product are produced. How many NADHs are made? How many ATPs are made? Are there any ATPs made at all through glycolysis or through pyruvate oxidation? And pay attention to the reactants, pay attention to the products, and pay attention to why the cycle is there in the first place. Why are those steps there, and how are they important to the overall process? Oxidative phosphorylation accounts for almost 90% of the ATP generated by cellular respiration. That's the purple box there, oxidative phosphorylation, most of it. But there is still a little bit of ATP made through glycolysis and through the citric acid cycle. For each molecule of glucose degraded to CO2 and water by respiration, the cell makes up approximately 32 molecules of ATP, all in all. Let's start with glycolysis. The word itself comes from splitting sugar splitting, breaking down glucose into two molecules of pyruvate. Glycolysis occurs in the cytoplasm, not in the mitochondria, and has two major phases. The energy investment phase, where we actually have to add a little bit of energy in, and then of course the second, which is the energy payoff phase, has to occur. Otherwise, what's the point of glycolysis in the first place? Glycolysis occurs whether you have CO2 or whether you don't have CO2. Glycolysis can always proceed forward. So what do I have to remember? What are the reactants of glycolysis? The steps, the products. Where does it occur? It occurs in the cytoplasm, outside of the mitochondria, and so here we go. This diagram shows that we start with the molecule glucose. Where does glucose come from, and how can we remember that it's the very first step of of this entire chapter? Well, in photosynthesis, the end product, glucose, ends up being the main reactant of cellular respiration. So we take the plant, first the plant makes the glucose through photosynthesis, and that glucose ends up being energy for itself, for the plant, but also we can consume it and undergo cellular respiration, which is breaking this glucose down and eventually making a whole bunch of ATP. So that way we can take that ATP and put it to use, making proteins and going about all the metabolic processes that occur in our bodies. Okay, you see how this diagram shows that in the different boxes there's a different steps: energy investment, energy payoff, and then of course you have the net down at the bottom. We actually use we actually have to start with a little bit of ATP. 2 ATP we actually use. And so we will break those 2 ATP into 2 ADP and 2 inorganic phosphate. Then in the then in the energy payoff phase, we actually make 4 ATP. We use up 2, we make 4. So what is the total amount, what is the net amount of ATP that we actually gain? Well, if we gained 4, but we actually started by using 2, we actually only have a net of 2 ATP. I hope that makes sense. Also, on top of ATP, we have another product, another important product. We have a product of 2 NADH as a net. And of course, we have 2 pyruvate. So the 3 important products in glycolysis are a net of 2 ATP, 2 pyruvate, and 2 NADH. And keep that molecule straight. It's not NADPH like in photosynthesis. This is NADH. Let's go through this step. Let's zoom in even more and go through the steps one by one to take a look. Here we have, on the very left-hand side, we have the glucose molecule. You see, C6H12O6. And we begin by going through step 1. We actually phosphorylate glucose. We take out a phosphate and we tack it on to the glucose molecule. So now we have glucose 6-phosphate. And you see that there in between 1 in step 1. Where we actually used up a little bit of ATP. How many? Well, just one here, and we tack on a phosphate. Where did the phosphate go? Why did it go from ATP to ADP? Where did that phosphate go? We tacked it on to the glucose molecule. Then we take that molecule, and what occurs in step 2? We go from glucose 6-phosphate to fructose 6-phosphate. So now what occurs? Through another enzyme, the molecule is converted yet just a little bit differently. And then from fructose 6-phosphate, we go to we gain yet another phosphate. Now we have fructose 1 and 6 biphosphate. So that means that we have methyl groups at 1 and 6, but notice we used up one more ATP. We used up the energy and we actually tacked on, we took off the phosphate from the ATP and we stuck it on the molecule, fructose 1,6 biphosphate. So there we've almost completed our energy and investment phase, and we've shown where have we actually used up, where we've actually consumed those 2 ATP molecules. All you got to do now is split up that fructose 1,6 biphosphate, cut that puppy in half, and you got yourselves 2 half molecules, 2 G3P molecules, each with 1 phosphate. So you see that all the carbons are accounted for, all the phosphates are accountable. You can even just ignore the dihydroxyacetone phosphate molecule. You can just say we have 2 of these G3P molecules, these 2 glyceraldehyde 3-phosphate molecules, which is really just fructose 1,6 biphosphate cut in half. That concludes step 4. Now, going from G3P to 1,3-biphosphoglycerate, we actually get out now a little bit of energy. We get out 2 NADHs. And then undergoing step 7, we get 2 more ATPs, and we end up with 3-phosphoglycerate. And eventually, we get out another 2 ATP, and we end up with 2 pyruvate. Let's just go back to step 1, and let's account for all that go into glycolysis and everything that comes out. We start with glucose. It's important that you remember that we start with the molecule glucose, C6H12O6. And halfway through glycolysis, we end up with 2 G3Ps, and in doing so, we actually consume 2 ATPs. We actually have to put in 2 ATPs. Then in our energy payoff phase, we end up gaining 2 NADH, 2 ATP, another 2 ATP, and finally we have 2 pyruvate. For a net of 2 pyruvate, 4 ATP, and 2 NADH. And that concludes the glycolysis step. So we look again at glycolysis, and what exactly occurs in glycolysis? Well, we start with 2 reactants, glucose and 2 ATP, and we get out. We've actually made 4 ATP, but because we've initially used 2, we actually have a net of 2, and then we have we've gotten out our 2 pyruvate and 2 NADH. So the arrow pointing down is showing us that we have 2 ATP. There's actually an arrow going up and going across to the right. That's showing the NADH that we've gained, our 2 products, the NADH product. And then finally there's an arrow going to the right from glycolysis, and that is depicting our pyruvate that is now the 2 pyruvate that are now going to enter the mitochondria and begin the pyruvate oxidation steps. So that's glycolysis. What are all the terms that I have to know in glycolysis? I would remember for sure glucose. Remember how many ATPs go in. I would remember that eventually halfway through we end up with 2 G3Ps and eventually we end up with 2 pyruvate using 1 glucose molecule, 2 pyruvate, 2 NADH, and a net of 2 ATP. Before the citric acid cycle can begin, pyruvate must be converted to acetyl coenzyme A, or acetyl-CoA, which links glycolysis to the citric acid cycle. This step is carried out by a multi-enzyme complex that catalyzes 3 reactions. All right, now this is showing just one of the steps. Now this is just 1 pyruvate. We have 2 pyruvate that will eventually enter, but for 1 glucose molecule, but we have, but this is showing just one of those steps. Here we go. We got pyruvate, and we take out CO2, 1 carbon and 2 oxygen, which is why it's in gray. You see CO2 in gray, and you see that part of pyruvate in gray. So CO2 comes off, and we actually gain NADH. You see how what enters is NAD+, but what exits, which is a product, if the arrow leaves the main arrow, then that means that's a product. NADH is a product, and we end up with acetyl-CoA. And of course, in order to do that, what enters is coenzyme A. The citric acid cycle, also called the Krebs cycle, completes the breakdown of pyruvate to CO2. The cycle oxidizes organic fuel derived from pyruvate, generating 1 ATP, 3 NADH, and 1 FADH2 per turn. This is the citric acid cycle. The citric acid cycle is we also can use the word carbon fixation. It's something similar to the Calvin cycle or the C3 cycle, but here we call it the Krebs cycle or the citric acid cycle. Now, let's take a look before we enter the Krebs cycle or the citric acid cycle. Let's take a look up at the top there once again. Here is our pyruvate molecule, and we've gained NADH, but because there's actually two pyruvates, we actually gain 2 NADH from pyruvate oxidation. Now, the Krebs cycle. What enters is acetyl-CoA, and the citric acid cycle has 8 steps, each catalyzed by a specific enzyme. The citric acid cycle, the acetyl group of acetyl-CoA joins the cycle by combining with oxaloacetate, forming citrate. So citrate is actually the first formed molecule, which is where the name comes from. Citric acid cycle is named for based on the first molecule that is made, which is called citrate. The next 7 steps decompose the citrate back into oxaloacetate, making the process a cycle. The NADH and FADH2 produced by the cycle relay electrons extracted from food to the electron transport chain. Let's look at the Krebs cycle or the citric acid cycle in detail. All right, so in comes acetyl-CoA, and the first molecule made is citrate. There it is. There it is. 1, 2, 3, 4, 5 carbons. We get out a little bit of water, and now we make isocitrate. We actually get out a little bit of NADH, and now we have alpha ketoglutarate. And through these steps, we actually get another NADH. We get a little bit of ATP. Now we're at succinate, and now we get out a little bit more FADH2, and finally the last, and a little bit more NADH, and finally the last molecule is called oxaloacetate. What I want you to remember from all these steps is that in doing so, through with one glucose molecule, we actually get out 6 NADH and 2 ATP. The citric acid cycle yields, or we gain, 6 NADH and 2 ATP. Now, that's not very much ATP. And in glycolysis, we don't actually get out very much ATP, but we do get a lot of energy. It's the 6 NADHs. We can't use those directly because ATP is the energy currency, the dollar bill that the body uses. But guess what? The 6 NADHs that comes out of the citric acid cycle actually enters the oxidative phosphorylation, and we end up getting a lot more ATP eventually. We just can't use that energy directly. The energy, the 6 NADH, enters another set of steps, and then eventually we can get out a whole bunch of ATP. Go to the left and remember the steps that we had just finished, the pyruvate oxidation. This diagram shows the energy that we've gained. We didn't actually gain any ATP here, but we actually gained 2 NADHs. And all this will eventually lead to the oxidative phosphorylation. Notice all the arrows coming off of glycolysis. we have 2 NADH. Pyruvate oxidation, we gain another 2 NADH. Citric acid cycle, we get 6 NADH and 2 FADH2s, and all these are energy-carrying molecules that are eventually dumped into the electron transport chain, and through chemiosmosis, we'll actually get out a whole bunch more ATP. So it's all like behind the scenes working, and it initially, or on face value, it doesn't really look like we're getting all that much ATP, but we're making a whole bunch of other energy-carrying molecules, NADH, all these different coenzymes, and eventually the energy is converted through oxidative phosphorylation into ATP. What do I have to know from the Krebs cycle? It's important to know how many energy-carrying molecules come out: 6 NADHs, 2 FADH2, and 2 ATP. You have to actually memorize those numbers. But also, I want you to remember that the first molecule formed is citrate, which is where the name citric acid cycle comes from. The first molecule formed is citrate. That's what I want you to get out of the citric acid cycle, or the Krebs cycle. That's the most important things. Following glycolysis and the citric acid cycle, NADH and FADH2 account for most of the energy extracted from food. These two electron carriers donate electrons to the electron transport chain now, which powers ATP synthesis through oxidative phosphorylation. Now we've gone through glycolysis, pyruvate oxidation, and the Krebs cycle, not yielding all that much ATP, but we're making a whole bunch of NADHs and FADH2, and finally all these products are dumped into oxidative phosphorylation to make a whole bunch of ATP. All these sets of purple blobs that you see here are actually multi-protein complexes. This is the electron transport chain. And we're going to see how this is similar to photosynthesis, and exactly what occurs at each of these steps. We're going to come back to this slide here and this slide here, but we're going to just talk about the electron transport chain here. So here now is the complete diagram of oxidative phosphorylation. You notice in the first set of steps we have electron transport chain, and then on the right we have another protein called ATP synthase, which altogether we see how the energy is converted from NADHs and FADH2s to ATP. This diagram shows the same idea as this diagram. Notice that this is this is inside of a mitochondria, and not just that, but it borders the inner wall of the mitochondria. So notice that the mitochondria has two membranes, and because it has two membranes, it actually creates two different spaces. The space that's in the very, very center, in the medulla, in the very center, we just call the matrix. But the space between the two plasma membranes is called the intermembrane space. This is important to note. So, because there are two membranes, we have an intermembrane space that's between those two membranes, and then the main area inside of the mitochondria we just call the matrix. Now, having said that, looking at this diagram now, we understand that on one side of this inner membrane must be the center of the mitochondria, the matrix, which is the top side here. The bottom side of this diagram represents the thin space between the two membranes. I hope that makes sense. Okay, that's important to know because we're going to create a high hydrogen concentration gradient, a high proton concentration gradient, and it's important to know where that high hydrogen concentration gradient occurs. Does that occur in between the two membranes or in the matrix? And the answer is in between the two membranes. Otherwise, we wouldn't even need a second membrane. Generally, organelles don't have two membranes, they just have one. This is why the mitochondria looks the way it looks, is because we have to create a high hydrogen concentration gradient in between the two membranes in the intermembrane space to help power ATP synthesis. Here's what we start with. We start with NADH. Notice the yellow, NADH. And there you have FADH2. Where did those come from? If we remember, those came from the citric acid cycle, pyruvate oxidation, and glycolysis. Now we finally can see and we can put to use all these molecules that have been made. We finally know where they go. They go into oxidative phosphorylation, specifically the electron transport chain. And here we have a set of proteins. These blue blobs are a protein complex, and inside of these proteins, the electrons are released from the energy carrying molecules and transported and passed along from one protein to the next. NADH we break up into NAD plus and hydrogen, and we also get out a couple of electrons. FADH2, the same idea. We put it in. You see, notice that the arrow is going into the molecule, and so it's a reactant. We can consider it a reactant, and the product would be FADH. But what about the whole? What is the important thing about that whole thing? Is that we actually get out electrons. We actually gain electrons out of those molecules. What do we need NADH for? What do we need FADH2 for? We need it to because they are carrying the electrons that we're going to put into the electron transport chain. And what then? Well, those electrons we actually get a little bit of energy from. The electron transport chain here in cellular respiration is a hydrogen pump. It pumps hydrogen against concentration gradient. It pumps hydrogens from the matrix to the intermembrane space, creating a high proton concentration gradient, just like the electron transport chain 2 in photosynthesis. Note that in photosynthesis, there were two ETCs, electron transport chain 1 and electron transport chain 2. 2 was actually the first one that occurs. Electron transport chain 2 was actually a hydrogen pump. And so the goal is the same. The hydrogens, then there are so many hydrogens in the intermembrane space that they're going to want to leave naturally. They're going to want to diffuse back out to equalize the amount of space between all the hydrogens. And they're able to. Where do they go? They end up flowing through the ATP synthase molecule on the right-hand side there, and through their kinetic energy, we are able to fuse ADP and inorganic phosphate together to make ATP. We're going to see in detail now, in just a second, how ATP synthase occurs. How ATP synthase actually does it in detail, does this making of this ATP. Okay, here's a summary of of cellular respiration. Technically, um, glycolysis is not cellular respiration. Cellular respiration occurs inside of the mitochondria. So all the things that happen inside the mitochondria we call cellular respiration, like oxidative pyruvate oxidation and oxidative phosphorylation. But glycolysis occurs in the cytosol outside of the mitochondria, and so um, we just call it glycolysis all by itself. Glycolysis can occur with oxygen and it can occur without oxygen. But cellular respiration can only occur only with oxygen. How is that possible? We haven't seen oxygen yet in this diagram at all, or in the mitochondria at all. So how is oxygen required in in the electron transport chain and in oxidative phosphorylation? And the answer is something has to happen to those electrons. And so the answer, as those electrons are passed from one protein to the next, from blue blob to blue blob all the way to the right, those electrons are now depleted, but they don't go anywhere. They actually accumulate at the end of the electron transport chain. And if they accumulate, they begin to back up. And if they back up, then the process can no longer move forward because the electrons are getting are just accumulating, and so the process will eventually stop. So we have to do something with those electrons. We got to get rid of those electrons, those depleted electrons. Where do we put them? We put them into the water molecules. We got hydrogens floating around. We got electrons, but we need one more component to make water, and that is oxygen. That's why oxygen is required in this entire cellular respiration business. We have electrons and we know where they come from. They come from breaking up FADH2 and NADH. We have hydrogens, those are just floating around in the matrix and the intermembrane space. But we can't form anything else, we can't form water without oxygen. So this is why oxygen is required. It's really, oxygen you can think of as an electron garbage bag, if you will. It picks up those electrons, and with hydrogens we make water, and that's why water is one of the products of cellular respiration. You could think of it as sweat, for example. So that is the main points of the electron transport chain. Now, the process of the hydrogens being pumped into the intermembrane space and back out into the matrix and back and forth and back and forth, this process is called chemiosmosis. Chemiosmosis is the process by which the electron transport chain uses energy from NADH and FADH2, pumping hydrogen into the intermembrane space, creating a high hydrogen concentration gradient, and the hydrogens flowing back into the matrix through ATP synthase, back and forth, is called chemiosmosis. That's the definition of chemiosmosis. And of course, in doing so, the final product is a whole bunch of ATPs, 28 in fact for just one glucose molecule. This diagram summarizes all the parts that we talked about. Glycolysis, you see the net 2 ATP on the right, 2 NADH, which we don't use right away, but the arrow goes down, 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 all the way to the ETC. And pyruvate oxidation, also we gain 2 NADHs, and that also gets dumped into the ETC chain. The Krebs cycle also makes 6 NADHs, and and also eventually gets dumped into the ETC chain, along with two FADH2s. And then finally, it's in the electron transport chain that we see um, all the energy in those NADHs eventually being converted into ATP for a final of 36 ATPs. And there is a reminder there that why oxygen is required, and that's because it picks up those accumulated electrons that um, that accumulate from all those energy-carrying molecules that will back up the cycle unless they're picked up by the oxygen, and you have the hydrogens finally making water. Now the last piece of the puzzle is fermentation. So, but before we get into fermentation, let's summarize one more time what's going on in as far as glycolysis and cellular respiration. If you remember, we begin. What are the reactants of glycolysis? We start with one glucose molecule, and we actually have to put in two ATPs. This is the energy investment phase. And in doing so, we end up getting a net of two ATPs, two NADHs, and two pyruvate. The pyruvate enter pyruvate oxidation, and we get out two NADHs, and the Krebs cycle, through the Krebs cycle or the citric acid cycle, we get out another 2 ATP, 6 NADHs, and 2 FADH2s, and all that energy gets put into the oxidative phosphorylation steps, in which we have the electron transport chain and chemiosmosis to get out considerably more ATP. Now, if you add these up, there's always this discrepancies, why 30 or 32 or 34 or 36, and there's theories on why. Maybe it's different kinds of animals. We're not exactly sure why, and it's actually not that critical for our purposes. The last piece, fermentation. Most cellular respiration requires O2 to produce ATP. Without oxygen, the electron transport chain will cease to operate. In that case, glycolysis couples with anaerobic respiration or fermentation to produce ATP. Let's take a look at this diagram here. We don't see fermentation here, but on this one we do. Notice fermentation, where does it occur? It doesn't occur inside the mitochondria, it occurs in the cytosol. But you have to start with glycolysis. Glycolysis makes 2-pyruvate, and if the pyruvate cannot enter the mitochondria, the pyruvate we can still make use of it, and it can enter the fermentation steps. If we have oxygen, the pyruvate can enter the mitochondria, and we can get out a lot of ATP. But if we don't have oxygen, we can still get some energy out of, we can still make use of the pyruvate and have the pyruvate enter fermentation. Let's look in detail at the steps of fermentation. On the left-hand side, We have glycolysis in one single step, from glucose to pyruvate, from one glucose molecule to 2 pyruvate. In doing so, if you recall, we actually gained a net of 2 ATP and 2 NADH and 2 pyruvate. Well, fermentation in one single step, we start with 2 pyruvate and we get out 2 ethanols and 2 CO2s. This fermentation that you see here does not occur in the human body, but does occur in other organisms like bacteria. Why would any organism want to do this if we're actually losing energy? Look at fermentation closely. We're actually consuming NADH and we're getting out NAD+. We're actually losing energy. Fermentation actually loses energy. Why would any organism want to do this? The answer is because NAD+ is the reactant, an important reactant for glycolysis. Move NAD+ over to the glycolysis step and you have the necessary reactants to start glycolysis over. And so if you do have NAD+ and you happen to have glucose, you then can move glycolysis forward and get out a little bit of ATP, 2 ATP to be exact. It's not that much energy, but it's enough to keep some organisms alive. This occurs not in us, but in other organisms like some bacteria. This fermentation that you see here does occur in the human body. Notice again, to begin with fermentation, we have 2 pyruvate, but we end up with 2 lactate. It's still the same amount of carbons as we see here, but it's just in a different form. Instead of ethanol and carbon dioxide, it ends up being lactate, and if you add an H, you can call it lactic acid. A lot of people will hear the name lactic acid, and that lactic acid is in fact what you feel when you run up the stairs and you feel that immediate burn. It's not the soreness the next day that we once thought. It's not the soreness the next day, oh, I feel the lactic acid in my legs. We found out that that's probably not what that pain is, but the immediate burn that we feel when we run up the stairs, that is because we are not clearing the lactate in the liver as fast as we're making it. We're not breathing in oxygen fast enough, and if we don't have enough oxygen, if the oxygen is not available, then the pyruvate cannot enter the mitochondria and make ATP. Instead, it undergoes fermentation, making lactate. So that is the summary of fermentation in the human body and also fermentation in other organisms like in yeast and so on. And that's the basics of what I want you to know for fermentation. Now, in this last slide, it's important to note that we don't get energy only from carbs, as we've been talking about glycolysis, glucose from carbohydrates, but we can also get energy by breaking down proteins into amino acids, and we can also get energy from fats by breaking it down into the fat parts, glycerol and the fatty acids, and also into the glycolysis steps or the acetyl-CoA steps. And that concludes the glycolysis, cellular respiration, and fermentation chapter.